Well, thank you. As Linda said, I'm a postdoc research associate here at Cornell and appreciated the invitation to share a little bit about what we're doing to advance some environmental DNA techniques. And it's a project that I started along with Jose Andres and David Lodge, who are also listed on this slide, um, along with Jesse Lepak, who's working with New York Sea Grant. And unfortunately, Jesse couldn't be with us today. He sends his apologies. He had originally planned to share a little bit about the Sea Grant network and work in general, but unfortunately has a Great Lakes fisheries commitment he had to be at today. So I'm going to be sharing a little bit about what he was going to mention too. And really, I should mention that a lot of the ideas for this research came from especially Jose, who has been doing a lot of population genetics work for a number of years in both terrestrial and aquatic systems. And then also, of course, David Lodge, who's been involved in environmental DNA work for about 10 years now, coming from an invasive species background. So my background, as Linda mentioned, is more fisheries ecology. And my plan for today is to provide kind of a general overview of first Sea Grant and then the project that we are working on at the moment, and share some details of that current project. We're about a third of the way into that process, which has mostly been lab focused so far on this project. And I mentioned that because our hope, and we've talked with Carrie and others about this too, is that this is kind of a heads up in some sense that this project is happening. And then later down the road when the project finishes, we're looking forward to sharing more about what we've learned and what some of the results are. But we wanted to make sure that those around the state who may be interested in knowing this is happening know it's happening, and that we can get feedback from all of you too. So that's going to be the third part of this talk, is just some feedback, and it doesn't have to necessarily be within this hour depending on our time, but feedback via email and in the future over the next weeks and months would be great from all of you too, in the way that basically this project can be most useful. So I'd like to mention a number of partners at first and collaborators, and a lot of the ideas for this project overall came through conversations with Carrie and the Invasive Species Research Institute, along with Rob Williams, who was involved pretty early on, and Carrie connected us to both him and to Dave Adams, Kathy McGlynn, Steve Pearson at DEC. And then Rob has then connected us further with the Finger Lakes Prism and Hillary, and then the Western New York Prism and Andrea. And they're involved, especially involved in some of the field sampling parts of this. And without their involvement, the ideas and the direction for this wouldn't be what it is. So I'm going to start out by just mentioning again a little bit about the Sea Grant system, because that's some of the background for this project especially. It was started in 1971 within New York State, and the Sea Grant College system is the formal name. It's a part of NOAA, which is under the Department of Commerce, actually. So it's part of the federal system that's a network of about 33 programs around the US, and it also includes Guam and Puerto Rico in the marine system as well. And the East, West, and Gulf Coasts are covered along with the Great Lakes, and actually along with Lake Champlain as well. And so I was first involved in Sea Grant through some of our Lake Champlain fisheries work several years ago. And they're basically modeled after the land grant college system and associated with land grant or research institutions around the country. And New York Sea Grant is a partnership between SUNY Stony Brook and Cornell University. And New York is actually the only program with both freshwater and marine responsibilities within it. And the main mission of Sea Grant is to conduct research, outreach, extension, and education with stakeholders, managers, and researchers, scientists around the state to address coastal issues. So around New York, that certainly includes some of the high water level issues, invasive species, harmful algal blooms, and then workforce development is kind of the category they've given, but development work with both managers and scientists around the state doing applied research and management. So part of this mission is then funding research in various capacities through the state. And the project that I'm going to be talking about started with a New York Sea Grant call for proposals several years ago. And that led us to discuss, okay, what ideas are out there that would be interesting to start studying. And at this point, David Lodge had just moved from being at Notre Dame, where he'd been for 30 years doing invasive species and environmental DNA work, to Cornell, and so was a new presence within the New York region. So we talked with a number of people, and Carrie especially mentioned that the Invasive Species Research Institute had conducted a survey in, I believe it was late 2017, late 2016, early 2017. And one of the research interests identified at that time was increased interest in the kind of development and application of environmental DNA for early detection. And so that was kind of the initial impetus for us pursuing this as a possible research project. So the title listed at the top of this slide is actually the title for the project overall. And the goal is to develop and test what is potentially a cheaper analysis method for environmental DNA. And that part is very much lab and technical lab based, and then test that method in Ontario and Lake Erie harbors. And it was that testing piece that especially came out of some of the Harbors Initiative ideas that Rob Williams especially had mentioned. So with that background, I'm going to talk now a little bit more about just kind of eDNA to start with. And again, not knowing the background of everybody on the call, I'm going to go through some things that some of you may know, but just some background for environmental DNA, and then talk more specifically about this project. So as you know, I don't need to go through this invasion process information in any detail, but I want to focus especially, and again, you don't need to read all the small print to the right here, but especially raise the focus that environmental DNA comes into play mostly at the introduction and establishment stages to be used as an early detection tool. And so it started around 2008, 2009 to be used in this way. And the research has continued from there. And the general framework is that DNA is present in the environment and can be extracted without actually isolating any of the organism that is present in that area. This figure here at the bottom, it's possible the arrow should be going the other way, but the general idea is that the distribution of biodiversity around is leading to a similar spatial distribution of aqueous eDNA within a particular region. But that is dependent on transport and degradation and processes along the way. So obviously there's a lot of advantages to this type of work potentially, but also there's a number of different ways where, or number of different tools that can be used traditionally to detect the presence of a certain species that we may or may not want to have in a certain area. And this line on this isn't really this isn't a data-driven plot, but just kind of a theoretical representation that in regions with a high density of a particular organism, they can be detected with relative ease, less effort, and a high detection probability. On the other hand, in areas with low density populations, obviously it takes a lot more effort and the detection probability is much less. And so again, this is just a theoretical representation here, but basically the message is that traditional field sampling methods are going to be more cost effective in a number of different scenarios at somewhere along this line where the effort involved has a certain detection probability, and that's related to the density of a certain organism within a particular area. At some point along that progression, environmental DNA sampling, depending on the costs and the focus of the effort, will become more effective at potentially detecting a species for a given amount of effort. And again, Linda mentioned I come somewhat from a fisheries background and community ecology fisheries background, and within the fisheries world, we sometimes talk about a catch or a detection per unit effort. And when some of the earlier studies of environmental DNA were done, there were analyses of Asian carp, in this case in Illinois, and this was with the Notre Dame research group at that point, looking at the catch per unit effort using electroshocking versus environmental DNA. So this was for silver and bighead carps and a comparison in that sense. So in this area where there's relatively high abundance of silver carp and bighead carp, it made sense mostly to use electroshocking as a detection method in that region. But in areas where they were less abundant, the catch per unit effort, person days in this sense, was much higher using an environmental DNA detection technique. So the background for this, as I mentioned, started around 2008 with this one paper study conducted in Europe with American bullfrog. And again, you don't need to read the titles of all of these, but it progressed from there, and the applications became more and more broad, and the techniques became more and more refined. So there were, again, studies conducted in streams. This was the study that was done with Notre Dame in the Illinois, Chicago area. Number of different broader biodiversity metrics, starting to get into the idea that we can use environmental DNA to detect whatever it is that's possibly in the water at a particular place. And then again, increasing both in the biodiversity monitoring, kind of conservation biology applications, and then also increasing the applications in the invasive species community. And that background is an indicator that there were a number of different advantages that everyone was trying to test out to this new method. For one, it's not destructive in the way that certain kinds of sampling techniques can be. We're just basically taking a water sample from a particular area. Second and thirdly, it's pretty sensitive. There's a higher detection probability, as I mentioned in those previous graphs, for a given amount of effort. Thirdly, it's possible to detect multiple species using potentially the same water sample and the same amount of effort that we would otherwise be using to focus on potentially one or fewer numbers of species. And that's worth mentioning that can also include pathogens and types of organisms that we might not otherwise be able to sample. And then also there's a reduced need for taxon-specific field training, which can be a big investment up front, and that is not necessarily needed in this type of sampling. And then finally, reduced permitting requirements are also an advantage in some case. And it's worth mentioning that that final point does not imply that open communication agreements aren't highly advised when doing this type of sampling, but just the process of some of the permitting requirements can be simplified when we're just taking a water sample. So there's a number of different applications for environmental DNA that have been used over time. Certainly species that are rare. So this can go for endangered species as well as invasive species, where there's a low density and we may or may not be sure whether they're present in a particular region. Secondly, species that are difficult to sample with traditional tools. Mentioned some of that earlier. And it also goes for microbial types of surveys, bacteria and other pathogens. Species which handling causes harm. I suppose that could be harm to both the person sampling or the species itself particularly. Habitats in which traditional tools are difficult to deploy. And that's kind of self-explanatory, a number of different reasons that could be the case. And then also where integrative samples are going to be useful. So there's been a few studies that have been conducted looking at the fact that environmental DNA in streams, for instance, can serve as kind of or the stream can serve as a kind of conveyor belt of biodiversity in a particular area. So the steps for conducting an environmental DNA sampling regime, relatively straightforward. Collect a water sample, and that can be done ideally in the field with a filtering that happens in the field, although sometimes it's possible, if needed, to take a water sample and then keep that cold for a certain amount of time, 16 hours or so, or less, and then filter it in a lab. Filter the water sample, as I mentioned, ideally that's done soon after the sample is collected so that the material doesn't degrade. And then extract all the DNA, and that's a step that's done potentially much later and in a lab. amplify the DNA, so make lots of copies of either all the DNA in a sample or particular pieces that the individual is interested in. And then visualize whether that DNA is present for a particular organism, if it's kind of a single species focus, or for a whole suite of organisms. So the more sensitive an assay, the greater the risk for false positives. And it is important to reduce the risk of contamination at every step, both in the field and in the laboratory. So we have a newer lab that was just built here with a number of individual clean rooms, especially for very sensitive analyses to make sure that contamination is minimized. And that's type of mentality is needed at each step along the way. So it's worth mentioning at this point potentially too some interpretations of environmental DNA results and implications for management. And we can conceptualize this in any number of ways, and it's been done in various types of publications, including this one, 2016. But we can kind of think of the weight of evidence for a species detection. At the bottom end of this, the weakest sort of evidence is just a limited number of eDNA detections, no way we think it could have gotten there, and also no physical specimens that have been collected. On the other end, we could have a number of eDNA detections on multiple occasions. We know there's a way it could have gotten there, and there's some type of capture record. And then, of course, we have anything in between on that spectrum. If we kind of think about this again in relation to the likelihood of secondary spread, we can then start to think about some of the implications for management, right? So if we have a species that we have a very weak amount of evidence that it exists in a particular area, and it seems to be in an area that is at low risk of future spread, it's unlikely that a response is as urgent as it would be if we have strong evidence, obviously, of a presence in an area and a high likelihood of secondary spread. Obviously, there's other characteristics of, say, the organism present and whether it's a high-risk species, and other pieces that come into play here. But we can start to think and rationalize different types of responses based on the amount of evidence that we have. And it's also worth mentioning just here that eDNA is obviously one tool in a toolbox that can be complemented by traditional sampling methods and other things we know about a place and a system. So I'm going to start to hone in a little bit more on what we're doing with this project especially, and do that by talking about a few different environmental DNA analysis techniques. So these are the techniques that we start to use once we've collected the water sample, filtered it, and then preserved that filter in a sample and refrigerated it or frozen it basically for a number of potentially weeks or months or years even. Once we have that sample then, back in the lab, we create lots of copies of the DNA that is in that water. First, of course, we extract the DNA from that filter material into a solution, but then eventually arrive at a place where we need to amplify those copies of DNA. The main way that's done at the moment is through quantitative polymerase chain reaction, quantitative qPCR. And generally when that's done, it's often done with a particular focus on one or maybe two species. And in order to do that, we end up using one sample or one subset of a sample in order to focus on one species. So it's a pretty active management approach in the sense that it's targeted. We're hoping to amplify the DNA of this particular species, bighead carp or tench, or something else that we're particularly interested in. Because we're just using it for one species, the relative cost is a little bit higher. The sensitivity is pretty high. We detect one species, and it's not extremely complicated, so the time required is kind of moderate. And it's a relatively simple bioinformatics process, so the information that comes out of these steps is pretty straightforward to understand and to process computationally. Another type of PCR method that is very similar to quantitative PCR in its application is digital droplet PCR. The main advantage to this is that it's a little bit more sensitive than quantitative PCR. Otherwise, we're still focusing on one species and kind of a moderate amount of time and lower complexity type of analysis. But the advantage is that both of these are targeted at a particular species. Again, bighead carp, tench, whatever it is that we're interested in. Another approach that you might have heard of, so these are basically a single species approach. We look to amplify the DNA from one species. On the other extreme is a metabarcoding approach, where we're looking to determine the diversity within an entire sample and sequence the genetic material from anything that's in that water. So we're not targeting anything in particular, which is more passive, and in terms of an invasive species approach is, again, passive. We're not interested in only looking at things that are of high risk potentially. Relatively low cost per species because there aren't as many, or we're spreading that cost over a wide variety of species. Lower sensitivity, a lot of species potentially detected, and then quite a bit of intraspecific variation that's possibly detected as well. So we were looking at these methods when we were thinking of ideas for this particular project and thinking that, well, it'd be good if we could have a method that was actively targeting species that were of interest, but was also able to do that with a focus on, say, 20 or 30 species at once, rather than just one at a time. And there was also at a more moderate cost than this type of method. So that's what we're working on with this particular project, is to develop a method where we're targeting about 30 species at once at a moderate cost per species, but a high sensitivity, similar sensitivity to these top two single species method, and also at a moderate level of informational, computational complexity and time required. So I'll just go through some of these steps of where we are at the moment with this process, some of the details of what that looks like. So the idea would be we have a couple ports or a couple places we're interested in, and we take an environmental DNA sample from that port, ideally a number of replicates, of course, from that particular area. We then amplify, create lots of copies of targeted species. So the emphasis here is that we're targeting particular species, but not just one. It could be 20, could be 30. And that's particularly done by using primers or markers, same thing, that are created to be specific to specific species, rather than one that is general and amplifies the DNA of all fish, for instance. Those are then marked. Again, this is getting into some of the primer and metabarcoding techniques here a little bit, which you don't need to remember all the details of, but we are marking those with a sample-specific tag, just so we can tell the samples apart. They're then pooled, and again, that's the importance of having a tag here, so we can pull out which samples are from which place. They're pooled and sequenced, and that reduces the sequence cost somewhat, is to be able to pool them that way. And then this is a difference from, again, the single species approach, is this sequencing part. When we're just targeting one species at a time, we create a lot of copies of that DNA and then can look at it on just a gel or other visualization just to see presence absence of that particular species. But in this case, we're targeting specific species, but then sequencing it also in order to determine what species it is that have shown up in the sample. So they're sequenced and then sorted by that barcode to get back to the sample ID, the port ID, and then sorted by forward primer. Could be sorted by other parts of the sequence that we obtain also. And that will get us back to the species level and the presence or absence of various species that were on our initial target list. So to amplify and create these species-specific primers, which is about the stage we are at right now with this project, for plants, we're using MATK and RBCL, which are two commonly used regions of the chloroplast genome. For animals, we're using cytochrome oxidase 1 and cytochrome B, which again are commonly used regions of the mitochondrial genome. Just to give you a sense of that and kind of a genetics reminder, both chloroplast and mitochondria have circular genomes. So this is generally what it is, and there's a number of genes that various research has mapped out on both mitochondrial and chloroplast genomes. And we're amplifying and targeting these particular regions of that genome. The main reason for looking at chloroplast and mitochondria too, there's a number of reasons, but they are more abundant than just a single genome for the nucleus and nuclear DNA. They're also inherited a bit differently than nuclear DNA. So I'll pause here a little bit, but this is the current target species list that we have for the project. And so when I mentioned we're interested in targeting 30 species, 20 to 30 species, that will then be amplified and detected potentially in future samples. These are the test species that are currently on our target list. These were developed mostly from starting with the Glancis Great Lakes watch list from Ontario and Erie especially, and then with some input from some of you on this call, for instance, and others with the U.S. Fish and Wildlife Service eDNA lab as well. But we're at the stage where we can still have some flexibility in this list. And so one thing we're interested in feedback on is whether there are any glaring omissions from this list or species on here that might not be of particular interest as a focus any longer. So I'll come back to this list in the future, a couple slides down the road here, but this is the current working list that we have. So as I mentioned, we're currently at the stage of developing primers. And just to give a quick kind of look under the hood of this process, what we end up doing is obtaining quite a bit of genetic material from NCBI, from GenBank, for each of the species that we're interested in. So it could be that there are 60 species, 60 sequences available for a particular species, and then we end up finding regions of the genome that can then be targeted for the development of a primer or marker for that particular species. Again, so you don't need to read all the details of this slide, but just to give a little bit of a visual, we really are working with chains of A, T, Cs, and Gs in text files and pulling out the material that can then be used to develop these types of primers. So the other piece I'd like to mention with this target species, so once we develop primers, I'll mention in the future that we're going to be testing some of this in the field in harbors in Lake Ontario and Lake Erie. But before we do that, we're also hoping to test the technique on a mock community, so to speak, in the lab. To do that, we're now in the process also of looking around for physical material from each of the species on this list. And so that's another piece of potential feedback that I'd be interested in if anyone happens to know of or have specimens of any of this material. It would be great to be in touch about that and potentially getting a very small amount of tissue from that sample. I'll come back to that and get that again in the future. So finally, just looking at our time here, as I mentioned, the final stage of this project, there's this big lab piece for methods development, and the secondary piece, not secondary in value, but second stage is testing this method on field samples. So to do that, we are focusing on 4 regions: Buffalo in Western New York, the Rochester Harbor area, the Oswego Harbor area, and the Clayton, New York area. So in each of these areas, the PRISM leaders in each of these areas have been patient with us as we've had some funding starts and stops and sporadic ability to conduct this sampling over the last year or so due to that funding. But we were able to collect samples from each of these areas with their help last July, August, and we're in the process of starting to plan some of that sampling for this upcoming growing season also. So that'll involve collecting about 15 samples, 15 replicates from around the region of these ports, of these harbors. And that involves using a 250 milliliter bottle, basically, and a single filter for that amount of liquid, a syringe to pump that sample through the filter, and then preserving that filter and bringing it back here to our lab to, at this point, refrigerate or freeze until we're able to use this newer detection analysis to process the sample. We can go into some of the details of that a little bit more if we have some time too. So I hope this has given you a little bit of an overview of the current project. Now I'm just going to focus again a little bit on our interest in having any feedback from all of you on this call and from anyone else that might be interested in that you're in touch with. In particular, the final piece, and not only as an output at the end, but throughout the process, Sea Grant has a great focus on staying engaged and extending and reaching out with whatever information we're gathering and being in touch with all of you and others around the state. And so we've talked about a future webinar, certainly as I mentioned at the end of the project, when we have more results. We've also talked a little bit more about, and with Carrie too, in-person workshops. We talked about workshops potentially associated with some type of conference. Also talked about written materials and or video type of materials also as outputs, either along the way as we work on this or at the end, or both. And so we'd be interested too in any feedback that either any of you have or that others have about the value of any of these types of ways of being engaged. Again, that could be in the near term while we're still working on this process, or it could be farther toward the end. So I've got both my email address and Jesse's email address here at the bottom. And we'd like if you could reach out to us and let us know any feedback you have. And this could be both about some of the technical steps that we're trying to push forward with this analysis and this project especially, or it could be more generally about how do we use eDNA in this particular place or time.